This unit is on translation. DNA has to first be replicated or copied. DNA always stays inside of the nucleus, except during cellular division. RNA's role is to transcribe DNA's message to form proteins. mRNA carries the genetic message from the DNA in the nucleus to the cytoplasm. The cytoplasm contains ribosomes, which use the mRNA to make proteins. mRNA is read 3 bases at a time. These 3 bases are called codons. Codons are read by transfer RNA molecules, also known as tRNA. Each tRNA molecule carries an amino acid on one side. The amino acids are the building blocks of proteins. On the other side of the tRNA molecule is an anticodon that matches with a particular mRNA codon. In this way, the cell accurately brings in exactly the amino acids that are coded for the DNA sequence. What we see here is a codon wheel. Once tRNA transcribes the mRNA message, you come up with this amino acid code. Looking at the codon wheel, there's 20 different amino acids. This chain of code will decipher which amino acid is going to code for a protein. Amino acids are read 3 codons at a time. So step 1, we have to separate your mRNA strand by the codons. If codons are read 3 at a time, you break up your mRNA message as follows, and you use the codon wheel. Starting with AUG, you start from the center of the codon wheel, looking at A. Then we have U, and then G. After the G, we see the amino acid of methionine. Looking at your next 3 bases, starts with a G, goes to a G, and then an A. We have glycine. Your next 3 bases, C, C, U, you have proline. Next 3 bases, U, U, and U, phenylalanine. The last 3 bases that we have are U, A, G. This is known as your stop codon. Putting these amino acids in order, we have methionine, glycine, proline, phenylalanine, and then we have our stop codon. On the codon wheel, stop codons are nucleotide triplets in mRNA that serve a key role in signaling the end of protein coding sequences. You can see that there's 3 stop codons, UAA, UAG, and UGA. AUG is a special codon. If AUG is found after a stop codon, it signifies a start codon for the next gene. If AUG is found in the middle of the sequence, AUG codes for the amino acid methionine. Any bases prior to a start codon or after a stop codon could be ignored. So let's look at the following mRNA strand. We have UCA, but we can't use UCA because we need to look for AUG. Once you find the AUG, then we start coding. And if you read this through, break it up 3 at a time, you have AUG, CUU, GGC, and UGA. We know that the trailing G will not be used. So what would the coordinating amino acid sequence be? You need to use your codon wheel to figure that out. Once you do so, we could break these codons up, these base pairs up by 3s, and we know that AUG codes for methionine, CUU codes for leucine, GGC codes for glycine, and then UGA is our stop codon. And then the transmission will stop, and then these 3 amino acids will get folded up into a protein. Now, the process just described is called translation. In translation, an mRNA message is turned into an amino acid chain. So what's happening during translation? The ribosome attached at the start codon, the AUG codon, and continues to move down the mRNA, bringing in tRNA molecules to create an amino acid chain until it reaches a stop codon. The tRNA molecules bring in amino acids to match the mRNA codons. Their amino acids bond together, creating a long string. The amino acids are bonded with a peptide bond, so the resulting chain is called a polypeptide chain. The chain will ultimately fold into a protein to be used by the cell. As you see, we have an overview of a cell. Inside the nucleus of the cell starts with DNA. DNA goes to get copied or replicated through DNA replication. Then mRNA, messenger RNA, goes and codes one side of the double helix strand. The mRNA is then able to leave the nucleus at this time and be able to be transcribed by tRNA codons. tRNA codons are going to create amino acids, then we'll create proteins. Here's an overview of how this works.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.